Good morning. It got really quiet. What well-behaved students, I know that you're really excited because this is a big day for Bentonville schools. And it's a big day and because of Congressman Womack. He had the opportunity to bring one of the world's best scientists to Arkansas, and he called me and said, I have 1 hour. And I said, we'll take it. And so I want you to give U.S. Congressman Womack a big hand for doing this for us today. And now I want to introduce our guest speaker. Dr. Francis Collins is an American physician geneticist noted for his discoveries of disease genes and his leadership of the Human Genome Project. He currently serves as the director of the National Institutes of Health in Maryland, enjoying bipartisan support from Washington within that role. Dr. Collins is a New York Times best-selling author, the recipient of hundreds of awards, including recognition from the U.S. World News and Report and Harvard University, and finally, an eager guitarist and motorcycle enthusiast. Scholars, teachers, and guests, please welcome Dr. Francis Collins. And if you'll join us in the seats, we have planned this morning for students to run the program because they do it best. I want to introduce the two ladies who will be facilitating the questions. Our students have put together some great questions today. Shorna Alum is a freshman at Bentonville West High School. If you'll join me up front. You know, her bio is pretty impressive as well. Shorna, come on up. She is the founder of the Bentonville West Biology Club. She also established the West. She established West as the only participant in the Biology Olympiad, the only Arkansas school participating in that, which is a rigorous nationwide exam devoted to biology. Thank you for joining us today and facilitating the questions. And then I want to introduce Elizabeth Jennings. She's a senior at Bentonville West High School. Elizabeth, come on up. She's the president of the Speed and Debate team. She is a National Merit finalist. Good to see you again. Being a National Merit finalist is a pretty big deal. She was an exchange student in Taiwan, and she is going to go to Abilene Christian University and pursue a physics degree. So ladies, I'm turning it over to you.
Right, thank you. I guess we'll sit down.
Yes. So, good morning, Dr. Collins.
Good morning. Good morning, Bentonville. Nice to see you all.
So on behalf of all of the teachers, faculty, and students of Bentonville Schools, I'd like to welcome you to Bentonville and say that we're all so excited to have a chance to learn from you today.
Thank you. You're most kind. I just got off an airplane coming from Atlanta. We've been at a meeting talking about what we're going to do about this terrible opioid crisis. But I was glad to be able to be here today at the kind invitation of Congressman Womack, who's been such a remarkable leader in the U.S. Congress supporting medical research, which is what NIH is all about. So a big shout out to him. And now in his role as chairman of the House Budget Committee, he has a lot to say about where our country is going. And he's a leader that you would want to have in that kind of position. He has vision, he has commitment. He's a smart guy. And he comes from a pretty cool place, Northwest Arkansas. There you go.
So again, sir, thank you so much for coming, both of you. And so to start off our symposium, Dr. Collins, could you please share with us a few of your early experiences in science?
Well, my education was a little unusual. I grew up on a small farm in the Shenandoah Valley of Virginia. And my parents were doing the 60s thing, except it wasn't the 60s yet. They lived on this small farm, and they thought that the county schools where we were not as good in terms of teaching as my mother would be. So she taught all of her 4 boys at home. And this was long before homeschooling was something that was much done. In fact, the truancy officer would regularly show up and wanted to know why these boys were not in school. But my dad, who happened to be a college professor on the side, convinced them that this was okay. And it was okay. It was more than okay, because my mother was a very gifted teacher, and she had the ability to make anything seem interesting, and when it stopped being interesting, change the subject to something else. So she could teach math, she could teach languages, she could teach history. She was a wonderful writer. She was a playwright. So I didn't go to school for those first 5 or 6 grades, and then we moved in town because my grandmother lived in town and had a stroke and needed somebody in the house. And there, the public schools were actually pretty good, kind of like this place where I gather your public schools are really remarkable. So I got the best of both worlds. I got started on the excitement about learning from my mother, but then I got to be in a public school where the courses were really superb. And what got me excited about science was a 10th grade chemistry teacher, John House, and he had the ability to tell you about scientific adventures that made you want to be part of them. And we had a lab course the very first day. This just stuck in my head. Very first day of this chemistry course, he gave each of us a sealed up black box about this big, and it had something inside it. And you weren't allowed to look until you'd come up with every kind of experiment that you might be able to do or even think about doing to tell you what was in there. Now, isn't that a perfect metaphor for what science is all about? You have a black box and you're trying to understand what's inside there. Mine was a candle, by the way, and I totally messed up and didn't know that's what it was, but it helped me understand why this was such a great way to spend your time. So I was hooked, and I thought, well, then I must have a plan to be a chemist. So I went off to college. I went to the University of Virginia, because that was my state university, and I majored in chemistry, and I loved that. By the way, I ignored biology because it seemed kind of messy. Well, it is kind of messy, but it's also really interesting. So after I finished college, I went to graduate school to get a PhD in physical chemistry, because that was what seemed interesting. And then I discovered, wow, there's stuff happening in life sciences that I totally missed, like DNA and RNA, and it's all information, and it has this satisfying set of principles, which I'd somehow not noticed earlier. And I realized, I think that's what's calling to me now. I want to work in the life science arena, build on everything I learned in chemistry and physics, but see how it could be applied to humans, and maybe even in a way that would help people who are suffering from illnesses. So here I was, sort of pretty far along in my training, and I did a big turn and went to medical school. I know, it seems like I was in school forever, and it did seem that way for a while, but ultimately it worked out for me really just the way I would hope it would, to have the chance to have some rigorous science training, to get into medicine, understand what happens in terms of clinical practice of medicine, and then, of course, how do you put that all together? Where's the area of science that is going to be satisfying in terms of principles and also applies to medicine? You got it, genetics. It had to be genetics. And that became my passion, and that provided all kinds of opportunities that came after that, including the Human Genome Project, which I'd never dreamed was even a possibility back when I was growing up. That was a long answer, so I should be more careful.
That was perfect. Thank you, sir.
So moving on from your early career.
Is that on?
So moving on from your early career, what journey led to your current position as NIH director?
Well, there was this passion that I had for genetics, and my laboratory, which was at that point at the University of Michigan, let's not talk about last night's basketball game right now, shall we? My lab was focused on trying to understand what was the DNA mutation that caused cystic fibrosis, one of the most common genetic disorders. In the 1980s, we didn't know much about this other than it was inherited, and it caused problems in the lungs, and it shortened lifespans, and it was pretty heartbreaking. And if it was inherited, that must mean that somewhere in that 3 billion base pair DNA genome, there must be a glitch that's responsible for that. Now, these days, that would not be such a hard problem to go and find the glitch, but back then, it was a bear because you had no tools to help you. The technologies weren't invented. the internet wasn't really invented. You didn't have the ability to just with a click of a mouse go and scan through the human genome. Nobody'd done that. So we had to work really hard. It took 5 years of lots of effort on parts of students and postdocs, and people got exhausted and burned out. But finally, in 1989, we did find the cause of cystic fibrosis. It was pretty amazing, by the way. You know, you have in your DNA instruction book, you have 3 billion letters, A, C, G, T, in a certain order. Just 3, a C, T, T. In one place in this gene on chromosome 7 is what causes cystic fibrosis. 3 out of 3 billion in that vulnerable spot. So that was a real needle in the haystack to find that, but we found it. And that was exciting, but it also was so incredibly painful to get to that if we were really going to do that for the thousands of diseases that we knew had hereditary contributions, we were never going to get there. We had to have a better map, a better set of technologies to scan through the genome. And that's how the Human Genome Project idea really got born, about how do we have an ability to do what we did for cystic fibrosis hundreds, thousands of times over, much faster and much more cheaply and quickly. So that was the Genome Project motivation, and I was pretty excited about it. And so some of you might be surprised to hear that actually at that point, the Genome Project was very controversial. And the majority of scientists in life science were opposed to it because they thought it just was impossible. You'd never be able to read that number of letters of the genome. And it would take too long, and it would probably cost more money than you had planned, and that would take money away from other things. And they also said, it's going to be so boring that nobody who's a good scientist will want to work on it. There were suggestions that prisoners ought to be assigned a certain number of base pairs, and if they did a good job of reading them out, they could be paroled. It was that kind of attitude that this was just really not interesting. What? Reading out the instruction book for human biology? You only do that once? That seemed like the most exciting science you could imagine. And so a bunch of us did get engaged in that, and our first leader of the Genome Project was none other than James Watson. You can't imagine a more rock star status kind of scientist to do this. This is James Watson, who with Francis Crick back in 1953 figured out that DNA is a double helix. He was still in his 20s at that point. Well, here he was signing up to lead the US part of the Human Genome Project, and we all signed up to work with him. It didn't turn out that well. Dr. Watson, for all of his scientific, amazing talent, also was one of the rudest human beings that's ever walked on the earth. And after he'd pretty much insulted everybody, including his boss, Bernadine Healy, who at that point was the director of the NIH, he was sent packing. Oh dear, here was the Genome Project just barely beginning to get off the ground. Think of it as a baby in the crib at this point, and we lost our leader. Now what happens? Well, my phone rang, and it was the director of the NIH calling to say, I want you to apply to come and be the director of the Genome Project. I thought that was crazy. I was really happy in Ann Arbor. I was seeing patients and talking to medical students about genetics and doing research, and that was what I thought my path would be. But here was this chance to actually do something really historic. And after debating and going back and forth and talking to my mother about it, by the way, my mother had an opinion here. She said, you know, you 4 boys, I'm proud of you. You've always been able to do whatever you set your mind to, but there was one thing that I never wanted any of my 4 boys to be, and that's a federal employee. So don't do this, because, of course, it meant going to NIH and becoming a federal employee. I rarely disobeyed my mother, but this time, after really thinking about what the opportunity was and realizing how much I would just hate myself 20 years later if I passed this up and it actually worked, I took the job. By the way, my first day as the director of the Human Genome Project was April the 4th. No, it was April the 3rd. April the 3rd, 1993. I believe that's exactly 25 years ago today. Whoa. So I guess that's why I got asked to do this. They needed somebody. They wanted somebody who both was interested in the science about how do you read out DNA sequence, and also somebody who could see the medical implications, and being a physician, that was maybe also an appeal. It was tough. Boy, I'll tell you, those first few years, I really didn't think we were going to succeed. We had so far to go to get up to this pace that had to be done. I mean, imagine what that scale is like. When I started as the director, people could get a PhD for reading out a few thousand letters of the DNA code, a PhD, and we had to read 3 billion. We had to get to the point where we're reading 1,000 letters every second in order to imagine that we could do this in the space of the last couple of years. It was breathtaking. But, you know, we recruited a lot of smart people. They got pretty fired up about it. 6 countries got involved. It was my job to be the field general and try to be sure everybody was pulling their weight, because you couldn't say you'd finished the genome if some parts of it weren't done yet. There were interesting times, conference calls at Friday at 11 o'clock in the morning. I'll never be the same, because that was always the time where everybody got on the phone to see what had happened that last week. But it worked. And what do you know? 2003, 2 years ahead of schedule, $400 million under budget. That made the Congress pretty happy. We were done. We did this. In exactly 50 years after Watson and Crick announced that double helix, we announced we have the human genome. April 25th, 2003. Pretty amazing. Now you asked, what about after that? So I stayed on as the director of the genome science effort, trying to figure out how to interpret this. Because if you had the genome in front of you right now, it wouldn't mean a whole lot. It would just be page after page after page of this 4-letter alphabet. You have to understand, where are the genes, what do they do, how do they get turned on or off in different tissues, what happens in disease, what about cancer, all this stuff. So that was another exciting 5 years. And then I was kind of restless. I kind of figured, like, maybe it's time to do something else, but I don't know what it is. So I stepped away. I became unemployed for a year. I wrote a book about personalized medicine. I tried to think about what my next calling might be. I was really interested in the interface between science and faith, and had the chance to talk to a lot of people about that, because I find those to be actually wonderfully complementary. And as somebody who became a Christian in my 20s, I was puzzled why people had such a sense that there was controversy there, because it didn't seem like there was to me. And then I got called and asked to come back to NIH, this time as the director. Now, the NIH is the largest supporter of biomedical research in the entire world. Thanks to the Congress just a couple of weeks ago, and again, a big hats off here to Chairman Womack. The budget for the NIH this year is $37 billion. That's a lot of money, and it's my job to oversee the 27 institutes and centers that make up this complicated organization, and be sure that every dollar of that gets spent to advance the cause of science and to help us understand both basics of how life works and how you can apply that to cure terrible diseases. And we're all passionate about that, and this is the most exciting time to see that happen. I thought after the last election that I would then do something else, because the NIH director has virtually always changed when there was a new president. But this time that didn't happen, and President Trump asked me to stay as the NIH director, so I am privileged to still be there and to still have a hand in steering this amazing ship of discovery to try to come up with those answers that are increasingly within reach. And what a privilege that is. It is an amazing experience to be given that opportunity. Another long answer.
Perfect. Thank you so much, Dr. Collins. So at this point in time, we'd like to invite some of our peers to come up on stage and ask their own questions. So if
Sure.
they would come up.
Great.
While they're coming up, let me explain to you why it's important from my perspective that any time the scientific community can discover the genesis of an issue that drives the cost of health care higher and there are many examples of it, it should automatically have the attention of the people who appropriate the dollars, the tax dollars, to support the research. For this reason, it is spending money today, maybe a few $billion, to, whether it's a moonshot or not, to find a discovery that could save trillions of dollars in the out years. That's what drives my interest in supporting the research that the NIH does, and it is precisely why I asked Dr. Collins to come to my district and to see some of the great things going on in the Northwest Arkansas area. So I look at it from purely a budget and appropriation standpoint. I think the money we give Dr. Collins is money well spent. It's going to improve your quality of life, and it's going to save the taxpayers a lot of money in the long run based on the work that they do. So that's my connection to it all as chairman of House Budget and as an appropriator and a vice chair of the subcommittee of appropriations that actually funds his organization, Labor, Health, and Human Services. So that's my involvement.
That's good.
Thank you so much, Congressman Womack and Dr. Collins, for coming here. If we could give them a short round of applause for that. And so our first student question for today is from Amira.
Hi, Amira.
Now, what grade?
My name is Amira Doherty. I'm from Old High Middle School and I'm in 6th grade. I have 2 questions I would like to ask you today. My first question, do you see gene therapy as a promising treatment for severe allergic disorders?
For severe allergic disorders, certainly gene therapy is bursting with potential right now. And we've seen, after 30 years of really hard work to try to get all the parts of this together to begin to see success, that is definitely happening. For severe allergic disorders, it would need to be a circumstance where there is a specific gene that's missing that, if replaced, could provide a benefit in that case. And there are examples like that. Gene therapy would be very appropriate. But sometimes severe allergic disorders are much more complicated, and we can't necessarily say, well, it's this gene or that gene. It's maybe 10 or 12 different genes that are interacting with the environment that gives rise to the allergy. So in that situation, it's not quite so straightforward. And in those situations, sometimes other things, like drugs, may turn out to be a better approach. But every condition has to have that kind of thinking applied to it. Where's the best chance that we have to block what seems to be an unhealthy situation? Maybe it's gene therapy, maybe it's drug therapy, maybe it's changing the environment, maybe it's changing health behavior, diets and exercises. We just have to understand conditions well enough to know where the opportunities are the best.
My second one, what steps need to be taken to implement?
Well, let me say, with gene therapy, the idea is that you want to replace a gene that's not working with one that is. And rather than just wave hands about it, let me tell you about a specific example that I saw when I was visiting Nationwide Children's Hospital in Columbus, Ohio, just about 10 days ago. There's a really terrible childhood disease called Spinal Muscular Atrophy, SMA. You all probably heard about ALS, and you know about the ALS challenge where all those people were pouring ice on themselves. Well, this is like ALS in a baby, and it's a genetic disorder. So these kids end up very floppy. Their muscles basically lose their tone, and ultimately they can't breathe because their breathing muscles don't work either. And without treatment, invariably, these kids make it to maybe a year and a month or 2, and that's it. Well, we know that a specific gene is defective in that situation. It's a gene that needs to make a protein in the spinal cord, which is involved in having those motor neurons that cause muscles to be able to move, and the motor neurons are dying out. So you could imagine how to do this. You want to put that normal copy of that gene into the right place. How do you do that? Well, you need a delivery system. You need a molecular version of a delivery truck that's going to go to the right location and present that gene and have it make up for what's missing. And in this case, it's a virus that's being used as the delivery truck, a virus called AAV. So you take the normal copy of the gene that's missing, you stitch it into that virus. The virus is otherwise benign. You give it in a simple infusion, just in an intravenous injection. And this virus is very clever, and it's been designed so that it actually goes to the spinal cord very efficiently and doesn't go a lot of other places. So I saw videos of kids who'd been treated this way. And remember, most of them don't even make it past about 15 months. I saw videos of kids 2 or 3 years old who are not just breathing, they're running around. One of them's on the monkey bars having a pretty good day. This is unbelievable. And this was a very early study. They just treated 15 kids, every single one of them still alive, and most of them looking really amazingly well. That's gene therapy, really where you want to get it to. And that gives us all confidence that we ought to be able to do that for lots of diseases. So yeah, got to get all the pieces together, though, to make it work.
Thank you, Dr. Collins. Our next question comes from Isabella.
Hi, I'm
Hi.
Isabella. I go to Fulbright, and I'm in 7th grade. And so here's my question: what difficulties during your research did you come across, or what really stumped you, and how did you overcome it?
Plenty of them, I'll tell you. It's probably easier to give an example. My research lab right now, because I keep a lab going at NIH, it keeps me anchored in the reality, and it's fun to be able to continue to teach graduate students and postdocs. We work on the rarest form of premature aging, a disease called Progeria. How many of you ever heard of Progeria? I'm just curious. Yeah, quite a lot of hands. So you may have seen a film called The Life According to Sam, an HBO film with the narrator being Sam Burns, who at that time was 15 and has Progeria. This disease causes your body to age at about 7 times the normal rate. And if you don't come up with some way to treat it, these kids die by age 12 or 13 of heart attacks or strokes, even though their mental development's entirely normal. If anything, I think they're unusually bright. So it's a heartbreaking disease to try to wrestle with. My lab discovered the cause of this, and this is just one letter out of 3 billion that's misplaced and in a very vulnerable spot, and it causes this condition. And we wanted to come up with a treatment for that would work. And so, being such a rare disease, you couldn't really get a company interested in developing a treatment. There are probably only a couple hundred kids in the whole world right now that have this condition. So we had to be creative and see, were there treatments that actually were developed for something else that might turn out to be right for this condition? And we hit on one, a drug that had been developed for cancer that did not work for cancer, but it had the right properties. And so we tried that first in a mouse, and then we tried that in the kids. And the good news was it clearly did provide benefit. These kids now live about 5 years longer than they were before the treatment. But the bad news is they still have progeria, and still by the time they're 19 or 20, they're in real trouble, and they do not end up surviving. So what to do? We tried a second trial where we added 2 other drugs to that. And this was the frustrating thing. It made total sense that these drugs would work, and it actually made them worse. And this is the reality of when you're doing medical research, that it is research. You don't know until you actually ask people to be your partners and to volunteer for a clinical trial, whether it's going to help them. You might have all the data that you could imagine from studying animals, and you can still be wrong. And in that case, we were wrong. We had to stop that trial quickly because it was clearly not helping. There's another bit of good news that we are now on the trail of a new approach, which is much more gene therapy in its strategy, which I think is going to turn out to be dramatically better than anything we have. And right now, the frustrating part of that is to get all the data together to convince the FDA that we're ready to do a human trial, because they want to be sure, especially after we had a bad experience, that we're not going to do something to hurt these kids. So it's all that. And research is not one of those things where everything goes well. In fact, I would say most labs that are doing really cutting-edge work, most of the time the experiments fail, because otherwise you wouldn't be doing anything very interesting. You're trying out a hypothesis. You're maybe designing a protocol to do something that's never been done before. Most of the time, it isn't going to work. But the trick is to learn from it, to figure out why it didn't work, and to then design the next experiment, which has a little bit better chance. That's the nature of it. Sometimes people get really frustrated and even feel this is sort of a personal defeat when your experiment doesn't work. I used to feel that way. And then I realized, no, that's the nature of the business. That's why science is what it is. That's how we make progress. And if you're really working in an interesting area, you should count on failing. If you don't fail very often, you're not doing anything very interesting. Keep that in mind when your lab experiments fail.
Of course, Dr. Collins, thank you. So Kendall provides our next question.
My name's Kendall. I'm from Washington Junior High. I'm in 8th grade. And where do you think genetics will take us in the future, especially with all the new technology we're receiving?
It's going to take us in a lot of interesting places. We've just been talking about gene therapy. We also, I think, have the chance to use what we're learning about genetics to practice better prevention, to help people stay healthy. That's been a dream that maybe is closer to coming true every day. In just a few weeks, you're going to see in the news media a big announcement. And that is we are going to invite 1 million Americans. Right now, you've got to be 18 or older, but pretty soon we'll open this up to all ages. 1 million Americans to take part in an unprecedented study of how you stay healthy or how to manage illness if it happens. So this is a study called All of Us. Get it? All of Us. And it will, for those 1 million of people, ask them to fill out all kinds of questionnaires about their health behaviors, to give a blood sample which will result in their complete DNA being determined, their genome sequenced. They will walk around with wearable sensors like the Fitbit that I have on, only something really fancier than that, that will keep track of what's happening to them. And their electronic health records will be available to researchers. With 1 million people following over the course of months, years, maybe decades, we are really going to find out how you could use genetics and the interaction of genetics with environment to help people stay healthy. And this would be called precision medicine, not just one size fits all, but precision for individuals to figure out what works best for each person. We don't know that very well right now, and we're going to find that out. And I'm just curious, if I asked you all out there, if I set up a booth back at the back of the auditorium when we get done here and said, here's a Q-tip, I just want you to swab your cheek and stick it in this tube, and I'll call you in a week and give you your complete human genome sequence. Would you take me up on it? Let me see a show of hands. How many people want their sequence? All right. Now let's think about it. How many people say, no, not for me? It's okay to say that, you know. You might want to know, wait a minute, who else is going to look at this? And will there be some chance that that information might get used against me in some way? Because maybe I'm at risk for some illness and somebody might want to decide to pass me over for an educational opportunity or a job or health insurance. Fortunately, we have protections against that kind of misuse in this country, and that's a great relief. And I suspect every one of you will end up having your complete genome determined. It'll be in your medical record. It'll be there when something happens where maybe you need a prescription and the doc is going to be able to say, oh, this is the wrong drug for you because of your DNA. Let's give you that one instead. That's coming, and it's coming pretty soon. Let's say over the next half decade, maybe decade. But for you, that's like a blink of an eye. So this is going to transform the practice of medicine.
Our next question comes from Monisby.
Hello, I'm Manasvi Koturi. I am a freshman at Bentonville West High School, and my question is, how do you collaborate with doctors to improve your research?
Well, we need them. So much of research now is not done by lonely individuals toiling away in small laboratories at night. It's done by teams. And certainly all the things that we're trying to do to bring science forward for medical benefits, we need doctors engaged in that who understand all of the ins and outs of medicine. So one of the things that we do at NIH is we invite teams to send their best ideas to us, and then we go put them through the most rigorous kind of peer review you can imagine, where other experts look at the proposal and decide whether it's worth funding. And then if they say yes, and it's really in the top 20% now, it's about the best we can do, then they get funded. So we bring teams together by the opportunity to get supported. Sometimes there's areas of research where we think there needs to be more activity, but we haven't been getting those applications, and then we put out an opportunity. Let's take the brain, for instance. By the way, this is another really exciting area. Between your ears, you have about 86 billion neurons. Each of them have about 1,000 connections, and they're doing amazing things right now. Some of them more amazing than others. I won't speculate about what your circuits are up to. But those circuits, which allow you to be sitting here absorbing this information and maybe clicking with something else you heard yesterday and bringing up a memory and all the things your brain can do, we're determined to figure that out. And there's a big project right now to do that. And to do that, we needed to bring engineers into this, and robotics experts, and especially computer scientists, and physicians and neuroscientists all work together. And that wouldn't just happen spontaneously. So then we put out an announcement. Hey, there is money here. If you want to work in this exciting area, get together with some other team members and tell us what you'd like to do. People sometimes say, oh, being the NIH director, you got all those scientists out there. They all have their independent ideas. This must be like herding cats. And I say, yes, it is like herding cats, but I know how to herd cats. You move their food. Think about it. And NIH has a lot of food, $37 billion worth of cat food, and we can really put that to good use when there is something like the Brain Initiative, or maybe cancer immunotherapy, or maybe regenerative medicine, where we want to get a lot of people to work together. And that's what we do. And when that happens, if there's a doctor out there that's interested in research, we've got something that they might want to take part in. And I hope many of you who are thinking about your own path, and I suspect in this room there must be quite a few who are thinking about maybe medical school someday. The most satisfying way, this is a little bit of a personal view, to be a physician is also to be a physician involved in research. It's great to take care of patients, but you might get a little tired of it after a while. You want something that's going to drive your own creativity, and that's what research can do. So I'm shamelessly advocating that some of you should become physician scientists, and I hope you do.
Next is John Blake.
I'm Blake Suter. I'm a 10th grader here at BHS, and I've heard a lot about antibiotic resistance.
Oh.
And I was wondering, what is the NIH doing right now to combat or research this resistance? And is there really a risk of like a large-scale failure across the spectrum of antibiotics?
It's a great question. How many of you have heard about this problem of antibiotic resistance? So you've been talking about it a lot, and you should. If you go to any major hospital and walk into the intensive care unit and find out what's happening there, a lot of the people who are there have serious infections. They might be blood infections, they might be pneumonia, they might be urinary tract infection. And somewhere in there, it would not be surprising there's somebody who has a bacterial organism that is resistant to all known antibiotics. And then you're in real trouble. So how do you help that person get better? You kind of have to tide them over until their body can fight it off. And sadly, that often doesn't work. So yeah, tens of thousands of people are dying every year of infections for which we don't really have good treatments. The problem is we haven't developed a lot of antibiotics in the last 20 or 30 years. There was a big outpouring of new antibiotics for a while, and then that stopped. It's what you call a market failure, because companies that might be developing antibiotics don't see this as likely to be profitable. Because if you were successful in coming up with the next generation of an antibiotic that is going to work in those patients who have otherwise resistant infections, you're not going to want to use it unless you have to, because you don't want that resistance to develop the next day. So it's going to have a very small market, and you're going to treat people for a short period of time, and it's going to be very hard for a company to recoup their investment. So what does that mean? That means NIH, funded by your taxpayer dollars, is the place that needs to step in here and take on this role of doing the discovery and the development of the newest set of antibiotics. So we are very invested in that space. In this most recent bill that you just heard about that happened 2 weeks ago, thanks to Congressman Womack and all the other folks that voted for it, there's an extra $50 million that NIH was given specifically for antimicrobial resistance. One of the things we're doing that's turning out to be pretty interesting is to depend on naturally occurring organisms that live in the soil as a source of new antibiotics. We have had a hard time in the past really taking that to the level we wanted to, because most of those organisms you can't grow in the lab, but they're there in the soil and they have DNA. So now by extracting DNA from soil samples and figuring out what microbes must have been there and what kinds of antibiotics they are making, we've started to discover whole new classes of antibiotics that are unlike anything we've ever tried and therefore should not have resistance. We have work to do. We have to then purify those, understand their chemistry, try them out in animal models to see if they're safe and effective, ultimately take those to human clinical trials. But we are on that path. We need to move as swiftly as possible because this is a major problem right now and we're kind of behind where we should be. But we will do our part. And I think companies do get interested if we've de-risked the project sufficiently so it looks like, hey, this is going to work, and it's just a matter of scaling up the trial and then manufacturing the compound, then it seems like it's worth it. If they have to start from scratch and the risks are too high, they haven't gotten involved.
So our next question is from Patrick.
My name is Patrick Morrison. I'm a 10th grader at Bentonville West High School. My question is, what is your advice to younger people aspiring to become scientists?
What a great question. Well, I think the most important thing is to try to find an opportunity to get directly involved in a research project. It's hard to get excited about science just by reading about it, although that can be exciting. It's hard to get excited about it just by sitting in a lecture and listening to somebody talk about it, although that can be exciting. I'm not putting that down. But the way to really find out if this is your calling is to have that chance to do something yourself. That's why we have science fairs, is to try to get that opportunity. Sometimes that's inspiring. Sometimes people haven't found it quite to be the thing that turns them on. So if you have the chance along the way to volunteer, even if it's not doing a lot in a research lab, but learning what goes on there and having a chance to have a little hands-on experience, that is the most important thing. If you have that inclination, find out what you might do in the summertime. At NIH, we have a summer intern program. We have 1,200 summer interns on our campus in Bethesda, Maryland every year. Many of them are high school students. And I sort of tracking what happens, I can't tell you how many times at the end of that summer, somebody will say to me, well, that did it. I really know this is what I want to do. I was thinking about it, but now I've had the chance to have this experience. I know this is my real calling. That's what I want to come up with. It's hard to get to that point without testing it out by having a real experience of that sort. And around here, I don't know what those experiences might be, but having heard about all the things that are happening in Northwest Arkansas, including a brand new children's hospital that I guess just opened like 10 days ago, there must be things going on that you could see what you could find out about in terms of volunteering in that way.
I'm going to ask our next question.
Okay.
So, do you think gene editing is ethical? And should we start this journey of gene editing, or is it best not to open this Pandora's box?
Okay, now we're going to get into the ethics stuff. I love this. How many of you know what gene editing is? Oh my, lots of attention here. Okay, so this has come along pretty quickly. At least it's come along in a practical way fairly quickly. We've talked about gene editing for a long time, but CRISPR-Cas, this remarkable enzyme system that came out of nowhere 3 or 4 years ago, has now made gene editing front page news. And the idea here is that you do now have the ability to take those 3 billion letters of the genome in a cell and change one of those letters rather precisely and not have a lot of risks of damaging other parts of the genome. And that sounds pretty powerful. I think the ethical question is, okay, on what cells are you doing this and from what species? If you want to do this on a mouse to create a mouse model of a human disease, well, we've been making mouse models. This is just a really efficient way to do it. So yeah, go to it. But if you want to modify a human embryo that you're going to end up turning into a baby, and you're changing the instruction book of that individual, even with benevolent intention, for me, that's one step too far. That says we are basically taking charge of our own biological future in a way that's never really been done before. And that means you're not only affecting that person, but all of their future offspring. And how often are we really sure we know what we're doing in human biology? Not always. In fact, most of the time. And suppose you did something you thought was beneficial, and now you've doomed that person and all of their future generations to a really bad outcome, and they didn't give your permission for that. Furthermore, I can't come up with a really compelling reason for medical purposes why we need to edit a human embryo. I could explain that further, but trust me, it's pretty hard to come up with examples where that is a necessary step to try to avoid a bad outcome for a genetic disease. The things that people worry about are using editing to actually create a superhuman species, maybe to make them better warriors or smarter mathematicians or whatever. And most of those scenarios are utterly unrealistic because the kinds of things you're talking about are not controlled by single genes at all. But I am excited about gene editing for humans, just not the embryo, okay? Put the embryo aside and let's say we really shouldn't be doing that right now. NIH will not support that research right now. But take a disease like sickle-cell anemia. It's a pretty common disease. There are 100,000 people in the United States who have this, and millions in other parts of the world where malaria has been frequent. You probably know this story that malaria is less severe in somebody who's a sickle cell trait, and so that trait has been allowed to reach a higher frequency because those kids survived. But if you get 2 copies of that sickle mutation, you have the disease, and that is a pretty challenging disease to deal with. You have these crises that are incredibly painful, and over the course of time that does damage to kidneys, to lungs, to heart, to bones. We need to cure this disease. And this is a disease where 1 letter out of 3 billion has been misspelled. You got a T instead of an A in that 6th codon of the beta globin gene on chromosome 11. That's just that 1 letter. That's all it takes. Well, why not apply gene editing in that situation? How would you do it? Well, like this. Where do your red blood cells get made? In your bone marrow. Do we know something about that? You bet we do. We could take out some of your bone marrow. I won't advise doing that to you today. And basically purify the stem cells in the bone marrow that make blood. Hematopoietic stem cells, they're called. We know how to purify those. Then take your gene editing system, your CRISPR-Cas, and go in and fix that mutation. You can check pretty quickly to make sure you did, and then take those stem cells that you fixed, expand them up to large numbers, and give them back to that patient with sickle cell disease after you've made a little space in the bone marrow for them to take up residence. That's a transplant, but it's not a donor transplant. It's an auto transplant. It's your own cells. You're not going to reject them, but they're fixed now. We have done all those steps in a mouse. It works great. There is no reason we can't do that in a human, and I'm going to be bold enough to say we can do this in the next 5 years. We should be able to cure this disease in 5 years using gene editing. So I get worried when people get exercised about gene editing and maybe it's not ethical. They're always thinking about the embryo part, and I agree, that's a place we should look at with great care. But there's a lot of things we can do with gene editing that don't involve embryos or what you'd call the germline, that involve treating the cells of somebody who has an illness to fix them. Kind of like the gene therapy thing we talked about with SMA, which is a different way to do that repair than gene editing, but it accomplishes the same goal. So I'm big on gene editing. NIH just announced a $190 million program to figure out how we could use gene editing for those thousands of genetic diseases where we know the DNA misspelling, and we ought to be able to come up with a way to fix it by this powerful strategy that's just kind of appeared. And I think in the next decade, some really cool stuff should happen. Maybe you'll all be part of that.
Thank you, Dr. Collins. I'm afraid we're close to our time today, but on behalf of all of the junior, middle, and high school students here, we'd like to say it's been an honor and a privilege listening to you speak today, and thank you so much for coming to Bentonville Schools.
Thank you for your great questions. Pleasure to be here. Thank you all. Thank you so much.
Thank you guys for being such a great audience today.